Hello everyone, my name is Barry Gallagher, a leading scientist, and today I'm here to talk about the Civil War and your body. Our main theme today will be about AIDS, but before we can begin, we must discuss a few concepts beforehand. I will first start on a microscopic level, beginning with the pathogen, and from that point, we can move on. This lecture will address all the assessment statements in 6.3 listed in your textbooks in order. Now you can all keep up. To begin, in our first assessment statement 6.3.1, a pathogen is any organism or virus that causes a disease. You can see in this example right here what a pathogen is. And these little buggers cause a lot of problems. In relation to our main theme of AIDS, one of these little pesky pathogens is what causes it all, and we'll address specifically how that happens later on. I'm going to use a lot of real-world examples to illustrate exactly what I mean for all you little simpletons out there as well. So be ready for that.
I'm so bored.
Let's move on to how to treat diseases caused by these pathogens. The most common way is to use antibiotics. These things are extremely effective against bacteria, but not viruses, and this is because antibiotics are substances or compounds that kill the growth of bacteria, and they do this by targeting the metabolic pathways of them prokaryotes. For example, prokaryotes contain 70S ribosomes in bacterial cell walls, while eukaryotic cells don't. So antibiotics can kill bacterial cells without harming humans. This means viruses don't get attacked either, since they lack those features too. Viruses do not carry out metabolic reactions themselves, but instead infect host cells and take over their cellular machinery. In order to deal with these, you have to treat them with specific antiviral agents that target specific features to the viruses instead, such as reverse transcriptase in retroviruses. Killing a virus with antibiotics is like killing a honey badger with the king cobra. It just don't work. Look for yourself.
Oh, now watch this. An antibody's up in the tree. Virus doesn't care. Virus does what it wants. When it's hungry, it eats.
That's a mean honey badger. However, our body has a few mechanisms to prevent the pathogens like the honey badger from entering. Our skin is our first line of defense, since it protects us from the environment and is made of dry, dead skin cells. It also contains biochemical defense agents like sebaceous glands that secrete chemicals which inhibit the growth of some bacteria. Our skin also releases acidic secretions to lower pH and prevent bacteria growth. Secondly, our mucous membranes in our bodies protect our internal structures. They are made of a thin region containing living surface cells that release fluids to wash away pathogens, and they contain biochemical defense agents like lysosomal enzymes that kill them. Mucus can be sticky and trap enzymes as well. Crazy. No, right? Let me show you an example with our good friend Mr. Turtle. Mr. Turtle and his shell show exactly what I mean.
This mean pathogen ain't gonna break through my hard defense membrane.
On the occasion when pathogens do enter our bodies, there are a few tricky things our bodies can do. Our skin and mucous membranes are referred to as the first line of defense, but once they are breached, the second line of defense goes into action. This line of defense is non-specific, since it does not differentiate between types of microorganisms and always invokes the same response. Examples include phagocytic leukocytes, inflammation, fever, and antimicrobial proteins. Phagocytic leukocytes, called macrophages, circulate in the blood but may move into body tissue in response to infection by concentrating at the sites of infection due to the release of chemicals from the damaged body cells. They can engulf pathogens when cellular extensions surround the pathogen and then fuse. The created vesicle may then fuse with the lysosome to digest the pathogen. This mechanism of endocytosis is called phagocytosis. The third line of defense are the specific defenses, coordinated by a type of leukocyte called lymphocytes. These can recognize and respond specifically to different types of microorganisms and have memory, which means they can respond more effectively upon reinfection, and this is specific immunity. Let's look at some lemmings to better our understanding.
The pathogens surge past the first line of defense, only to be stopped by the intimidating, daunting task of battling the macrophages in the bloodstream. They go for it anyways.
Crazy little fellas. Now we'll talk about antibodies and antigens. An antigen is a substance that the body recognizes as foreign, and this recognition can evoke an immune response, while an antibody is a protein produced by a certain white blood cell, usually B lymphocytes or plasma cells, in response to an antigen. Antibodies are Y-shaped molecules, and the ends of the arms are where the antigens bind, and these areas are called the variable regions. And these will typically differ between each antibody. Each type of antibody will recognize a unique antigenic fragment, making this interaction specific. Now let's talk about how antibodies are produced with regard to Assessment Statement 6.3.6. Don't forget, students, we have covered all assessment statements from 6.3.1 to 6.3.5 up to this point. The B lymphocytes, usually called B cells, are antibody-producing cells that develop in the bone marrow to produce a highly specific antibody that recognizes one type of antigen. When the macrophages find a pathogen, they digest it and present the antigenic fragments on their surface to helper T lymphocytes, usually called T cells. These cells activate the appropriate B cell, which divides and differentiates into shorter-lived plasma cells that produce massive quantities of antibody, like a huge industrial power. A small proportion of B cell clones develop into memory cells, which may survive for years, providing long-term immunity. Alright, class, now that we have developed a solid understanding of the immune system, let's talk about AIDS. AIDS is caused by the human immunodeficiency virus called HIV, which is a retrovirus that infects helper T cells. Reverse transcriptase, which is an enzyme used to generate complementary DNA from RNA, allows viral DNA to be produced from its RNA code, which is integrated into the host cell's genome. After a number of years of inactivity, during which the infected T cells have continually reproduced, creating a mass amount of them, the virus becomes active and begins to spread, which destroys the T cells in the process. This is why AIDS takes so long to develop. This results in lower immunity, as antibody production is compromised, and the individual is now susceptible to opportunistic infections from a weakened immune system. AIDS stands for Acquired Immunodeficiency Syndrome, and it is a collection of the symptoms and infections caused by the destruction of the immune system by HIVs. AIDS is the final stage when observable symptoms develop. And HIV is caused by the transmission of bodily fluids usually. But there are a few ways to reduce the spread. For example, using contraceptives during sex. AIDS is a far more serious disease than many of you might think. Not only does it kill you, but it also kills everything around you socially. People with HIV may be stigmatized or discriminated against, and it can result in an increased number of orphans, and it could also be taxing to a country's welfare resources. Poverty may also increase the transmission of AIDS due to poor education and high costs of treatment, and all this creates a moral obligation for assistance from wealthier countries, which is also taxing for many of them. And so I end my lecture here, but before you go, here's a little clip I made to show you all exactly what is going on for your own benefit. Your homework is to make a voiceover over a video of all the things we've learned today.